Welcome back, dear students, to the trail of microbiology scientists. And today we are going to talk about uh, the environmental microbiology scientists. Previously, we were mentioning basically most of the scientists today were of the medical field. And now, today, we are coming across some scientists who played an important role in the soil microbiology as well as the environmental microbiology. And the first person is Sergei N. Vinogradsky. Sergei Vinogradsky, as a picture, you can see close to him, there are certain, close to his picture, there is different columns of, or some tubes are there, which have been found to be multicolored. Now, we'll see what is that. If we go to look into Vinogradsky, he is a soil microbiologist and he found out that bacteria could oxidize iron and sulfur as ammonia to obtain energy. He also found out that bacteria can convert carbon dioxide into organic matter like the photosynthetic organisms will do. That is, the photosynthetic role of bacteria was also being was being depicted by Vinogradsky. Normally, we say that carbon dioxide is converted into organic matter by plants. But the role of microbes or bacteria in the conversion of carbon dioxide into organic matter was being explained by Vinogradsky. And he also discovered anaerobic nitrogen fixing bacteria, Clostridium pastörianum, and the various interactions of microbes in cycles within the natural environments was also being studied by Vinogradsky, and he developed a column called the Vinogradsky's column, and the decomposition of cellulose was also being analyzed by Sergei and Vinogradsky. Now, when I go to talk about all these discoveries, we can see that this scientist is a man who dedicated his life for a non-medical area of microbiology. Usually, we consider microorganisms associated with diseases. But we know that microorganisms are not only associated with diseases, but for the ecosystem to function properly, for the biogeochemical cycles to undergo properly, or for the proper recycling of the nutrients, microorganisms do play an important role. And right from the start of photosynthesis, bacteria do play a role. That was explained by Sergei Vinogradsky. And when you have the plant material which is made up of cellulose, that has to be decomposed. There also bacteria is being found to be involved. And when you need to have a nitrogen fixation, even anaerobic nitrogen fixing bacteria are present, like Clostridium pastörianum, which was discovered by Vinogradsky. And there also do exist iron and sulfur and ammonia oxidizing bacteria, which will use these as a source of energy. Now, we mentioned a column is being made for this man, or credited a type of column is being credited to the name of Vinogradsky. So let's what's that? We'll check that. Now, what is this Vinogradsky's column? It mainly consists of a transparent cylinder which was being prepared to analyze the microbial life by observing how the metabolism of microorganisms evolve over time in a stratified layer. What is a stratified layer? A stratified layer is different layers which are being arranged one over the other. And to produce a ecosystem which was similar to under the soil, what he did is he took a column and he mixed some soil with some water and all that, and he let them settle. And when this, of course, the mud was also being mixed with certain sulfates and carbonate salts, as well as cellulose and other organics were also being mixed at the bottom, and it was allowed to settle in a column. So what happens? The soil itself will arrange in different layers within the column, and as a result, when the different layers have been formed, different zones of oxygen based zones also do appear within the column. In the topmost region, there would be aerobic zone. Then after that, below that, we will have a micro aerophilic zone. Then less anaerobic zone. Anaerobic zone. Anaerobic means without oxygen. More anaerobic. And finally, you will have a total anaerobic zone at the bottom also. So based upon the oxygen based distribution, different zones of will be formed within the soil. Now this column is depicting in fact a typical soil atmosphere which could occur within in the earth's surface. Vinogradsky is trying to depict the different layers which could be possible, which could be formed, which could be possibly formed by different microorganisms in a soil flora, in a soil ecosystem. So how did he prepare this? He took a glass cylinder and into that the sediment is placed, and then he overlaid them with salt water. Either you can do it like that, or and within the top of the column it is sealed, and much of the cylinder becomes anoxic in such a way, and sometimes the addition of shredded newspaper is also being done. Now, if you're not taking salt water, you can mix it with sulfate and carbonate and salts and cellulose or other organic matters, depending upon what procedure you are doing. So, when you are arranging, when you look into this, you can see that multiple colors, as I in the previous slide I had told you, the oxygen-based differentiation, stratification of layers happen, and based upon the different levels of oxygen, different microbes get stratified at different layers. At the highly anoxic conditions, you can see that the fermentation of cellulose is being carried out, and fermentation products are being formed, and that is mainly being done by Clostridium. And this fermentation products along with sulfate is being converted into sulfide by another bacteria called the desulfovibrio. So in this anoxic environment, what happens? You can find anaerobic Clostridium as well as sulfite utilizing bacteria which produces sulfide, that is desulfovibrio will produce over here. And the H2S which has been formed over here is then again diffused out on top. And that can be utilized by certain bacteria called the chlorobium. And when chlorobium is formed, a green color zone will be formed. Then when above that, when chromatium grows, a red color zone will be formed. And above that, you can see that various photoheterotrophs like rhodospirillum and rhodopseudomonas will be formed, and they will give a characteristic rust colored zone. And above that, we have an oxygen dominated mud light brown area where algae, aerobic sulfur oxidizing microorganisms such as Beggetova, Thiobacillus and Thiothrax will grow. And on the topmost layer, you can find photosynthetic bacteria called the cyanobacteria, as well as certain diatoms could also be found. So based upon the oxygen based distribution within this column, you can find different bacteria are being found to be associated into different layers of the column. And Vinogradsky was trying to depict the distribution of microorganisms in a soil based ecosystem based upon the oxygen levels. And as I mentioned earlier, the cellulose is degraded into the fermentation products, and then that is being utilized by what? Desulfovibrio, and H2S which has been formed, it gets diffused, and that is being utilized by chlorobium and chromatium. So depending upon what is using, you have different colors, and above that region, what happens? You have the organisms like rhodospirillum and rhodopseudomonas. They are the purple non-sulfur bacteria, and they use organic matter as the electron donor under anoxic conditions and function in a zone where the sulfide level is being found to be lower. Now, both oxygen and H2S may be present higher in the column, allowing specially adapted organisms to function. So you can have chemolithotropes like Beggiatova and Thiothryx. Now, above here, now above this region, either you can have H2S or you can also have oxygen also. So if you're having H2S, basically, H2S utilizing organisms like Beggiatova and Thiothryx and all that will be H2S is used by Thiothryx and Thiobacillus and all that. And oxygen can also be utilized in the upper regions by the diatoms and the cyanobacteria. So the main contribution of Vinogradsky is all these. That is, he produced the Vinogradsky column, studied the decomposition of cellulose, the interaction of microorganisms in the soil ecosystem was being studied by Vinogradsky. Now, the next scientist which I would like to talk about today is Martinus W. Beijerinck. He made the fundamental principles of microbial ecology, and he is considered one of the founders of virology and the environmental microbiology. The concept of the occurrence of viruses till then was not known, but the concept of that was being put forward by Martinus Beijerinck. He discovered the aerobic nitrogen fixing bacteria Acetobacter and the root nodules associated rhizobium was also being discovered by Beijerinck. Another organism called the sulfate reducing bacteria is credited to Beijerinck, and you can see that another technique called the enrichment culture technique was being discovered by Beijerinck. Now, what is the enrichment culture technique? That is, in a normal environment or in a all the organisms will grow, but sometimes when you have some specialized organisms, like now, for example, plastic degrading organisms and all that, if you want to isolate them, you have to enrich that particular thing. That is, you have to give some extra nutrients so that what even the least amount of bacteria will be uplifted and they will be promoted to grow. That is referred to as an enrichment culture technique, and it plays an important role in soil microbiology as well as in microbial ecology. And it was Beijerinck and Vinogradsky who together developed the enrichment culture technique. And Beijerinck is considered to be one of the founders in virology, and in 1898 he published the results on the filtration experiments demonstrating that tobacco mosaic disease is caused by an infectious agent which is being smaller than bacteria. We know that viruses are non-filterable substances. That is, even on filtration, they will be what? You cannot retain a virus. They will pass through the filters also. So, the occurrence or the association of viruses with tobacco mosaic disease was being established by Martinus W. Beijerinck. So with this, I guess we come to an end of this session or depicting the trail of microbiology scientists. And today we had discussed two main scientists, Vinogradsky and Beijerinck. Hope so it is clear to you. Thank you for now.